The quokka, Cetonix brachius, is a small wallaby classified as vulnerable and only found in southern Western Australia. Quokkas aren't seen very often because of their low density, secretive behaviour, and occupying inaccessible habitat. Because of these characteristics, there is not a defined population estimate for them. The aim of this study was to use conventional methods for population estimation to calibrate a rapid survey technique for the quokka in the southern forests of Western Australia. The methods used included the Little O rapid survey technique and the capture mark release practice. The rapid survey technique is designed to provide a qualitative measure of relative abundance, which is conveyed as low, medium, or high. For this study, fecal pellets, runnels, track counts, and sightings were used. The second practice is capture mark release, which gives a reliable population number. This technique was used as an indicator of true abundance to later be compared to. The survey area was a 10,000 square kilometre region of forest. The study was performed in autumn, so it was drier and the vegetation was lessened, which concentrated the individuals to water sources. The rapid survey technique used 137 habitats, which defined the quokkas to creek beds and swamps. In these habitats, a subset of 12 sites were selected for the capture mark release technique. This involved a trapping web at each site, with the central axis of each trailing a creek bed. The traps used were Thomas softball traps, made with shade cloth stretched over a steel frame. Runnel, track, and pellet fecal counts were carried out at each site 10 days before the trapping began. All fresh pellets were collected daily to avoid recounting the same specimens. Track counts were completed near the entrances and along the runnels, and physically observed quokkas were also noted. Capture mark release data for each 12 sites were used in the MARC program to calculate the population abundance. Each category was significantly different, with low at 1.25, medium at 4.75, and high at 8.5 individuals. This can be seen in the graph. There were two main faults in the study. The pellet removal system may have affected the animals' movements, as they could be motivated to defecate where there are previous pellets. Trained observers were also needed to be used in the field, as others may not be able to determine between the quokka and the western brush wallaby species. As the fecal pellet count was the strongest contributor to this rapid survey technique, it needs to have minimal errors. From these results, it was concluded that the whole rapid survey technique could be shortened just to the fecal pellet count. This is because it had the strongest relationship with the population abundance calculated through the MARC program. If this is undertaken, the first accurate estimate of quokka abundance can be established in the southern forests of Western Australia. Thank you.